So, um, just before I start properly, I need to start with the Creative Commons license. This was inspired and adapted from a presentation from the past from Ruth Eiserling. Um, oh yes, before I start again, I hope everyone have downloaded the big file that I asked yesterday. If you didn't, start doing it now. Um, and let's go. So, uh, today we are going to talk about an experiment on graphs, which is a network of nodes and edges, right? Um, we will show how to create a new custom uh, app from a custom graph from scratch in Cytoscape, and we'll also talk about some third-party Cytoscape apps. So there is, sorry. So networks are really impressive. Usually, whenever you make a network, people will be like, "Oh my God, that's so cool!" And like this is particularly important for example, when you're doing poster presentations or when you're even a paper. If it has a network, it gives a certain aura of success on it. This is something that is like valuable for people. Uh, it is harder to make a good network than to make a network, though. So oftentimes you see figures like this that look really impressive until you notice that it's really hard to make sense of it, right? Oftentimes you see these super complex figures. Hello, I think someone entered here. Sorry, and it is like unless you really know what you're doing, you create just a complicated mess that is really hard to understand what's going on at all. Um, and you can do networks on many things. Recently, not recently, but this decade, people were talking about the 7 degrees of separation that in social networks, if you know a person who knows a person who knows a person, you can reach anyone in the world in 7 degrees only, which is an approximation, right? But still really impressive, right? You can get 99% of the population in 7 degrees. Um, and then people did some fun interactions on it. So I don't know if how many of you heard about the Bacon Erdos degrees of separation. Someone did the Erdos degrees of separation. Erdos is a famous mathematician. Um, who I don't know if he's alive or he died recently, but he's a mathematician from this century that was very prolific and he collaborated with many people. So you can, if you published a paper with Erdos, you are Erdos 1, and if you publish a paper with a person who published a paper with Erdos, you are Erdos 2, and so on. You can create a network of publishing, and you can use the references to reconstruct this network, and then you can get your Erdos number, which is how far away in publications you are from this mathematician specifically. And then people started doing the same things on IMDb for actors and people who work in movies. So if you are on um, credits screen with Kevin Bacon, you are Bacon 1. And then if you are on a credit screen with someone who is on a credit screens with Bacon 1, you are a Bacon 2. And then if you add those two together, you have a Bacon Erdos number. The physicist Neil deGrasse Tyson has one of the lowest Bacon Erdos numbers because he was on several TV things. So he has a Bacon number that is sufficiently small. And because he is a physicist, he also has an Erdos number. So he has like a 4 or 5 or something like this. I tried calculating this for myself. If I consider some undergraduate some works from undergraduate cinema students. I participated in some of those, so I would get an Erdos number of like 10. Um, Anyways, this is something that I thought was worth mentioning because it's a very weird thing that people do. And if once you know that it exists, you see lots of people talking about it on the internet. Um, so networks are an important way of visualizing data. This slide was more or less how it was 2 years ago. I wanted you to know this because I will do a comparison later. But this is an example that they gave showing how networks can be used to reduce complexity. So you have here about 200 pathways or something. Like it's really hard to put 200 pathways in in a table, but if you have here on a figure, you can more or less organize them. So they're way more efficient, right? They are great for data integration. So you can have many dimensions going on here. So here you have the edge colors, the edge thickness, the node shape, the node color, and also size. So you have here 5 different qualities shown on the same figure. So this is a very powerful tool. And it's somehow intuitive. Like you look at it and you can quickly understand that this guy, whatever it is someone important, and that these guys, whatever they are similar to each other because they are all connected to each other. So you can quickly look at a figure like this and get information quicker and more efficiently than you would get on a heat map. And then the changes that I made to this is that everything is only true if done properly. So this is a real figure that someone sent me. They were like, "Oh, look at this data that I found. How cool is it?" And you can't see anything. Uh, this is the greatest zoom that I can have on the figure. If you try to zoom in more, to try to read these labels, you can't because it's just pixels. Um, I don't know what to say about it. You cannot say anything about it. It's so confusing. Like you have some circles and some scattered nodes. What's going on here? So it can only reduce complexity if used properly. It is only more efficient than tables if used properly, and it's only great if used properly. So we need to be careful with what we do here. And in all examples that we will use, I will be reducing the number of nodes that we can handle. So sometimes you see, like in this cover of nature here, I believe these are all species in the tree of life. And we have here like more than 1,000 points. And you can see some patterns overall, but you cannot distinguish individual points. So it's not really useful. Here, for example, you can have labels for all the points, and then it becomes more useful. If someone really likes like a protein specific, they can try to look for it and find here, oh yes, I am studying SARS-CoV, and I want to know the interactions between the S protein and the this network, so I can look here for the S protein. Um, sorry, is the N protein. But here, the N protein is here, and you can see which are the proteins that interact with it, right? So, but here you cannot. So we can use these networks to represent many different types of data. We will be focusing mostly on the interaction of genes or proteins, right? As discussed yesterday, or sometimes we will have connections of pathways as well. And we will show both of those. Um, and one thing that we will be talking on uh, superficially is some characteristics that a network can have. So in this case, for example, we have right in the middle MKRN1. This protein is clearly what we call a hub. So all of the connections kind of converge to it. So if you want to go from any random node, let's say this one, to any other random nodes, let's say this one, and you want to create a path to it, you will most likely pass the hub. So there are several ways of measuring how much of a hub a certain node is. And one of the most intuitive ways is if you want to go from any point A to any point B, how often you pass through this hub. So this is what they call the betweenness centrality. So how much this node is in between other nodes. So you create thousands of different paths on the network connecting two different nodes, and then you see what is the ratio of these paths that pass through a certain point. So every node in the pathway has a betweenness number. There are other ways of measuring this too. Betweenness is the best one, in my opinion, for when you have a simple network in which the edges are they don't have a particular quality that you're interested in. Sometimes, like in this case here, you'd see that you have different thicknesses of edges. So some connections are more special than others. So then you need to take these weights into account, and then you have a connectivity measurement. The connectivity measurement is similar to it, but you will have to count every edge has like a cost or a distance that is inversely proportional to its width. And you need to do some normalization procedure to take everything into account. But it's basically the same concept with this adaptation for the witness as well. Um, so I will be talking a lot about these hub genes, but there are other qualities that you might also see around, like the local connectivity. So when you have a cluster and you just want to talk about the connectivity in this cluster and not in the whole network, and other features like this. Um, we will be using Cytoscape. There are other tools to make graphs and nodes, but this is by far the most popular one, and it is it's a very powerful tool. So I can't think of a node that can't be made on Cytoscape. It has like a billion citations, of course, and there are many apps that were built on it. So you can download these add-ons that will make it more complex, and we will use some of those. Um, the most intuitive, in my opinion, networks that we should do are these protein-protein interactional networks. As I was saying yesterday when I was talking about the different sources of pathways, like GO, for example, several of those were electronically annotated from these high throughput experiments in which you have many proteins that are connected to each other. And then you can try to create these paths in which you have one protein that interacts with a protein, and then another protein that interacts with another protein. So you create this indirect path between those two, and then you can start to see some structure appearing. So you have several clusters, and then you can try to presume that these clusters have a certain function in common, and then you predicted some pathways. Um, there is the big file that I ask you guys to download is from BioGrid, and this is a huge protein-protein interaction network. So they will have this enormous list of all of the proteins that interact with each other, and they have some qualities on the side. So there are extra columns showing the type of evidence that they have for these interactions, and sometimes they have other information as well. Um, so you can take any list of genes from your experiments. So just like we took any list of genes from mutations or from expression or from any other type of experiment like methylation, I don't know. If you have a list of genes, you can make a pathway enrichment analysis. If you have a list of genes, you can put them and you can compare them with an existing protein-protein interaction network, and then you can put your genes in this network. Um, let me see if I'm not forgetting anything. Oh, yes. So in this example, you can add also another dimensions to the network. So in this case here, the color shows the fold change. So I had a bunch of genes that were differentially expressed. I selected a threshold to only put those that are differentially expressed significantly. And then we can color them by fold change. So I can see that some genes are having verse correlations with other genes. So for example, uh, if LMBTL3 is a suppressor of expression of other genes, then it makes sense that this is reduced while all the other genes that it interacts with are increased because it is a suppressor. So if you have less suppression, you have more of everyone else. So it's very common for you to see these contrasts here of contrasting colors. Um, And you can also clearly see here that there are some nodes that are more important than others because they connect with many other proteins. The size here is proportional to the number of the edges. So you see that these hub genes becomes larger. This is very intuitive. This makes people able to look at a figure and instantly recognize the main players, to say. And I also changed the shapes here. So the more significant genes are triangles and the less significant genes are circles. Um, this is a way you can do things. Um, so this is another example. And then here the squares are kinases and the circles are the targets of the kinases. So kinases are a particular family of enzymes that will put a phosphorylation group into other proteins. So they bind to other proteins and they append phosphate with three oxygens, and this will change because the phosphate grabs the electrons from around it. It will change the conformation of the protein whenever you do this. So several proteins have this like on and off switch. That is a site that when phosphorylated, it will change the conformation of the protein. Then it will start acting in a different way or stop whatever reaction it was doing, for example. And this is a very common feature on many proteins. And in this case here, this is a study in which people were analyzing how the COVID virus affects these phosphorylation networks. So instead of affecting many different genes of a cell to change its cell cycle to be always producing new viruses and stop replication, uh, it just affects one kinase that will phosphorylate many other proteins. So you can kind of control this pathway regulator. So with a few targets, you can completely change the way that the cell behaves. And in this case here, you have these protein-protein interactions between kinases and their targets. And you can see here that some kinases have lots of connections with other targets. And the colors here are when which amino acid is responsible for this change. So sometimes you have, for example, a mutation that will change one amino acid, and then it will make the kinase work or not. Uh, all right. All right. Um, Connie sent a message talking more about the Erdos number here on the link. So these are many ways you can build networks, and some examples here. These are the networks for genes, sorry. And then I believe that one of the most sought after things that people want to do in this course is to do these networks using pathways. So these are very powerful. You can have, for example, I mentioned several times before that sometimes you can cluster pathways that are similar to discern them. So this here is an example from enrichment map in which you have the size of the nodes here is proportional to the size of the pathway. And then the edges show how well it is overlapping with other pathways. So microtubule cytoskeleton has a huge overlap with microtubule cytoskeleton organization and microtubule-based movement. You can't see here if the overlap is total or partial, for example, but it is still something that we can show. And then here on the color, you're showing if this is more upregulated or downregulated on average. So you take the average fold change of the genes that belong to this pathway, and you create a color for the node. This is how it should look like. For example, if we take the results from, I believe this figure I made from the pathways that we did yesterday. And there is one important thing here is that you see that you have a very dense network of edges because there are some genes that are very ubiquitous and they appear in almost all pathways. Um, so there is this slider whenever you open in the program. There's a slider you can choose to have more nodes or less nodes, and this will be based on the Jacquard coefficient. So if you have too many nodes, you have a blob. And once you have less nodes, you can start to see this blob taking shape and becoming like two different clusters, for example. And then you can see this, there's something like a path that the more important edges make. And what is the Jacquard coefficient? It is a clever thing, like it is a ratio of the union versus the intersection. And this why might seem arbitrary at first, but I will go with you under it. If you have a very big pathway and a small pathway that is inside of it, the small pathway has a 100% overlap with the big pathway. But the big pathway has only a 1% overlap, let's say, with the small pathway, right? So whenever you're creating an edge here, this edge isn't directed. So the relationship between the small pathway and the big pathway, you need to give a number to it that will represent the width of the path of the edge. And you can't have a different number for the big one and the small one. So you need to find a measurement that shows overlap that is kind of normalized between the overlap between the big pathway and the small pathway and the small pathway versus the big pathway. And this is where the Jacquard index comes from. You take the union of the pathways, so it's like the total area that they cover, and then you divide it by the intersection. So in this case, you have like an intermediate value that will show this relationship very well. So this is a ratio, so it's bound between 0 and 1 because the intersection will always be smaller than the union. Jacquard coefficients of 1 means that they are exactly the same thing. And 0 means that they don't overlap at all. And this is the original blob. If you move the slider a little bit, you create something like this. And this is something that, for example, I created this imaginary line here in the middle. So if you twist these two pathways, these two clusters around, you can start seeing something that makes more sense. So this is something that I just took these nodes and twisted them. And then we can see here that there are these two clusters and they are connected by T cell activation and differentiation. So you can clearly see some important themes going on here. You can see, again, the hub nodes here and these nodes that don't have particularly many edges, but they're still in between a lot. And if we have time today, this is something that I separated to the end. We will have also these figures in which each node is a pie chart, and then we can combine, for example, several different experiments here. So, for example, we were working with the comparison between two subtypes of pancreatic tumors, right? One thing that we could do, for example, is instead of comparing subtype A versus subtype B, we could have control A versus tumor A and control B versus tumor B. And then we have like two different comparisons with two different path enrichment analysis. And then we can combine them into a two-color graph. Like this one has many more colors, but we can do it with any number, right? Oh yes, and one thing that you might notice sometimes we've been ignoring this. Sometimes we have nodes for each of the different, names for each of the different nodes. And sometimes we have these blobs without names, and then we have a circle around it with a name on top. This is using a tool called AutoAnnotate. It's one of those add-on tools that Cytoscape has, or apps. You can download it apart, and then it will basically look at clusters. There's like a threshold that you can say to have like big clusters or small clusters. And then they will pick words from these names that are more different than the rest. So if we look at here, for example, you have regulation of cell-cell adhesion, leukocyte cell-cell adhesion, cell-cell adhesion, T-cell activation. So regulation is something that isn't that important because there is a regulation on the other side as well. So this word would be omitted. But cell-cell is something that happens a lot here and doesn't happen a lot on the other side. And on the other side, you have lots of lymphocyte things and leukocyte things, but there are a few leukocytes and lymphocytes here as well. So they will probably have differentiation as a smart word here. So AutoNotate will basically create word clouds for different clusters, and then they will take words that better represent these clusters. So here you will have things that are more or less tossed words. So in this case here, for example, you have APC DNA mitotic. So random words that appear more often in this cluster than on the other one. And in these final figures that we see in publications, people use AutoNotate to create these circles and to show the word cloud, but then they have to manually select which words will be shown in each circle. Because, for example, here you have dopamine signaling. Not necessarily AutoNotate knows that one is a object and the other one is quality. So it doesn't know the order each thing should go. So you need to kind of manually curate all of those. And again, there is some decisions that you need to take here. Sometimes you have nonsensical things mixed together because, for example, in these long clusters here, maybe you had something on the one side and something on the other side. Then you need to make some choices to make sure that it all makes sense in the end. All right. So now we will continue with the interactive part. I decided to reduce a little bit the talk to have more time for you guys to have hand-on experience. And so I want everyone to open Cytoscape. I hope you all have installed it beforehand, right? We downloaded the protein-protein interaction network from BioGrid. I sent you just a link for you to download, but it comes from this website. You can see here that they have many types of tables that you can download. I selected those that have the tab tree ending. This just means that it is a tab separated table, and the tab tree has more columns than the tab two. And again, they have several classifications. I guess that I asked you guys to download a file called organism, in which they have separated tables for different species. And then we took the human one. This file is very, very big. And if the previous file was crashing on your computers, this one would definitely crash even in R, probably. So what I did was to open this in chunks. So I sent you guys a script with it. Let's exchange here the window that is being shared. So this is the script that I sent you guys. I could I want you guys to follow this with me to create some of the files that we will be using. So here in the beginning, I will just be opening the file that we used before. This is the dseq2 results. And I'm using this function to split the entries ID from the gene, which we showed yesterday. There are different functions that can do the same thing. I like using this one. So I can run it and it's not finding the file because I'm in the wrong folder. Sorry. All right. Now it works. This is something that happens with everyone. Um, and then I ordered this table by p-value, and I selected the top 500 ones. As I said, I don't want to make a huge network with a billion genes, or 2000, for example, because it would get too messy. So let's take 500. 500 is a decently large number to put in a figure. As I said, sometimes you need more than a table. If you made a table with 500 genes, it will be impossible to read. But a graph with 500 nodes is doable. So I selected those. I selected here their IDs separately. And then I will open this file here in chunks. So instead of opening the file at once, I have here a file connection that's called. So it is just a pointer that will show in R where the file is in the disk. And then I can use this read lines function that has a number next to it. So here I read one line. This is the header. I will separate the header. And then while these chunks have not size 0, I will be reading more chunks. So here I'm using read table, and then chunk size here I've set it to 1000, but you can put like 10,000 or 100,000. It depends on how powerful is your computer. But you can read it in chunks. So you read the table, and then you split, you rename the columns. You can filter these chunks. So in this case, instead of opening the whole table, I am just opening the lines that will be relevant for us. So if we go back here to the slides, if I want to make a figure like this one, in which I have two genes being connected and both of them are in my data set. So all of the genes that you can see here are differentially expressed. So I'm only interested in edges that connect two genes that are differentially expressed. So then when we go to the code here, I had a list of interest genes that are in the top 500. And then I'm only taking genes where the ID is in my list of A and the ID of B is also in the list. Oh, yes. Let me run this to get the header. And then, oh yes, I said that here print, just for you guys to see what it looks like. So this is not the file itself, it's a pointer to the file. So it has like a description of where it is, what type of file it is. It's a text file. If you have read or write permissions. So this is not the file itself, it's a pointer to the file. And once we read the chunk of length 1, we have a header. And we can see, we can see here on the header how it looks like. It's a tab separated one. So when we use string split to separate it by tabs, we can see what are the column names here. And then we have here gene A, gene B, by interest gene, biogrid, name. So they have several columns showing A connects with B on different languages. And then they have some explanations on the source, the author, like from which paper did it came, right? And there are some other terms and IDs and qualities of these edges that we might look into later. So I'm using interest gene interactor A and interest gene interactor B to filter those. So here, the chunk will only show things that are in one list, and the other column is also in this list. And then we will save these chunks into a list. So we declare the list here in the top, right? So when we do this, it should take like a minute or so. And we will have a list with everything organized. Are you guys following with me? Do you have any doubts? I can't see everyone's faces. Now I can, sorry. So any comments while this is running? Oh, yes. It gives us this error because when it tries to read the last chunk, it doesn't have 1,000 lines. So you have like an incomplete chunk. Um, so the last chunk has like 439 lines. That's normal. But if we look at these results lists, we see that the first term is a table that has 37 lines. And then, oh, sorry, one line. The second chunk is another one with one line. Because it's such a small chunk, they are usually having one line only, but they don't. Some of them have a longer one. Um, if we do it like this, we will get the number of rows in each of the items of the list. And oh, I need to use apply maybe. I don't want to run it again because it took like 2 minutes. Um, so you can see here that some of them have like 4 rows, some of them have 8 rows, most of them have 1 row, but it's not like I just got unlucky. If we have here the number, actually, this might have more digits. The number 10, for example, it will have a length of 4. Um, so we can use do call to combine them all into a huge table. That won't be so huge because we're only looking at these 500 genes, right? And then it should have like more than 500 because several genes will have more than one edge. So it will have 700 connections. So each gene has on average a little bit more than one, like 1.5 connection. With just the column names, and then we have a final table. We can sort them. So here I have like the minimum and the maximum of each column. And this works even if the number of the strings. If you have like the max between ABC and CBA, it will say that CBA is the maximum one because it's later in the alphabetical list. So this is very cool. And then we just remove the duplicates, and then we will have a slightly smaller table. Let me do it again, just in case. 563. And then we can write it. And then we have this file that we can open on Cytoscape. So this is Cytoscape. I hope you all have it open. Um, Let me open here the slides as well. I really want all of you guys now to do exactly as I do. So we will start loading the file here. So we go to import, and then we choose network from file. So this will be creating a whole new network from a certain file. And then we have here on the outputs that we just created. Uh, Sorry, I'm having Zoom with your faces in front of the image, so it's not moving. Uh, so we'll be opening the file that we just created, mini biogrid. And then we have to select here that one of the columns is a source node and the other of the columns is the target node. Therefore, it will create these nodes that are named 1201448, and it will be connected to the one named 3853. So we start like this. And then we've created this monster here. Um, huh. Let me just see one thing here. Hmm. I was expecting it to have more edges because I probably changed some things in my pipeline in the middle. Um, so when I selected 500, just the top 500 genes, maybe I was too picky, and then I selected too few ones. But it created these things where you don't have many connections between them. Um, I don't know what I did wrong then. So this is what I was expecting to see. This is what is on the slides. Uh, but here I already have the names, so I changed the ordering somehow. Um, I changed the slides yesterday to make things more organized, and then I complicated some things. Let me just go again to see if I did something wrong here. Use the ones that was on the zip folder just because it looks nicer. In the file that I did here, I cut it too many edges, so we don't have too many of these connections between one gene and the other. Um, so we have this network that is kind of sparse here. So we have several disjointed nodes. Um, okay. Um, I will not lose more time with this. I did this whole thing of the chunks because I wanted to show you guys how to deal with complicated files that don't fit inside in your RAM. But let's not lose too much time because since yesterday we were missing so much time, I would just send this file again. All right. So I sent a file on Slack. And again, just to be sure, we import table from a network from file. And then we selected the one that we just downloaded, the mini biogrid. And then we have one column being the source node and the other column being the target node. And then we give OK, and then it should build this network. Um, is this working for you guys? Cool. Uh, oh, message. Yes, it works. Amazing. Um, so here we have the names already, and the names were like if we didn't have the names for some reason, or if we want to add more qualities to it, we can also import instead of network will create a new one, right? We just import a table from file. And then on this table from file, we can have, for example, this. Yes, I forgot to save this thing here. Uh, with write table, and then hmm, sorry, I was trying to organize the code to create like one that is incomplete for you guys to complete and one that is already done. And then I accidentally removed some stuff. But the idea is that we wanted to have this table that has the different express genes. I'm doing something wrong with the right table here. Yes, I'm was doing something wrong with the right table here. All right. Now we have this table that I saved as CSV, but it should be a TSV that has the ID separated. So then we have a column called gene name and a column called gene ID. Um, and something's wrong here. Um, I think I was doing something wrong with my right table function. Huh. All right, now it worked. Always an opportunity to learn from our mistakes, right? So what was happening is that I was selecting columns 1 and 2, and in final result, column 1 was this biogrid interaction ID that is just a number. And entries genes are also just a number. So I had two IDs that were just a number. So it wasn't easy to see what mistake I was making because I had this column 1 with numbers, column 2 with numbers, and I was trying to match column 1 and 2, but they were different things. This is why they weren't matching at all. And I was having this disjointed nodes. When I changed it to get the proper columns, now it worked. So on the final code here, I have the name of the column instead of column 2 and column 3. This is the more proper way, and it is the more proper way because it is harder to make mistakes if you have the names of the things here. Um, so yeah, careful with entries IDs because they are just numbers. Anyways, so now everyone has networks. And for now, this is a monster. This is really hard to understand anything going on here. And we are going to add more dimensions to this plot. So we go to import, and then we go to table from file. And then we have these DSIC results 2.tsv that I created on that. Uh, Okay. Oh, yes, exactly. Um, so we can add this as well. And this will be the file that we've seen before so many times, right? Uh, Here we are importing this data to an existing collection. So we could create for like a new table or something like this. This is the name of the collection. And then it will be imported as node columns, which means we're giving some quality to the nodes. So here in the end, we have entries ID and name. So the key here is entries ID, which means that it will match the lines from this column to the previous table. Our previous table of edges had entries IDs on both sides. So then the key should be entries ID. And when we click OK, it will load. And then we see here at the bottom a bunch of new columns showing up. I don't know if you all have these things in the bottom, but this is the node table. So the way Cytoscape is structured, you have in the middle your main image, right? On the corner here, you have these options. So you have like network, style, things like this. We will show them later. And on the bottom, you have these tables. So edge table is a list of all the edges, right? This is the first thing that we did was create this one, which automatically created the first columns of the nodes table. And then when we imported
Sorry, my edge table is empty.
your edge table is empty,
Yeah.
but you have something in your image.
Yes, I have this blue network.
So you can
Node
roll.
table, I have all the columns, but edge
Yeah.
table, there is like nothing.
So zoom out, and if you click around, you move the image, right? But if
Mm-hmm.
you press Shift, you create a square, and then you can select everything.
Okay.
When you select everything, um, something appears on your table.
Not in the edge table.
On the network table, do you see anything?
No, both tables are empty. I only see columns
When
in
you
the
go to
node
network
table.
here, you have it blue on my mini BioGrid 6.
Yes,
Mm. You are on the web or on the normal program?
the the software.
Okay. Um, on the nodes table is also empty.
No, that's like yours.
Okay. Hmm. Let's continue for now and
Sure,
see if you have a problem later.
sure.
So when you select a node, it will show just the node that you selected. So this is why I asked you to zoom out and select everything, because maybe you had something selected and then it wasn't showing on the edges. I don't know. But, well, for now, we have all of these tables. So we included all of these tables to the nodes, and the nodes have, so we have here the p-value and things like this, and we have the gene name as well. So when you go to style, here in the corner, you have all of these qualities that you can put to your network. So you can go here to label, uh, and sometimes you have to click here, and then it will show that there is a column called name that is the name of the nodes. But you can select here to be gene name, and then the name of the nodes becomes the gene names. So it was just uh, name, so it was numbers, and then you can choose to gene name. Okay, so this is the first step of fixing things. We can go to fill color then, and then we select a column called log fold change. And then on the mapping type, uh, so previously we had these pass through, which is if the name is CPS1, then you write CPS1. Sometimes you have discrete, which is something like if it is positive, paint it red. If it's negative, paint it blue. But we are going to put continuous here. This will create a color gradient. And then all of our genes will be colored according to this gradient. So it will, we're using the log2 fold change column. So this is good. We don't need to change that. If you wanted to, you can double click here and then you can, for example, change a little bit the contrast. You can also change the colors here. You double click on the triangle on the top and you could put some funky colors here. And then your graph is weird. Let's put it back because it's unsettling to have a purple and red plot.
How did we get the color palette? Sorry, I missed that step.
You click on the small triangle here. You see
Okay. Yeah.
when you double click it, it should open the palette.
Okay, got it. Thanks.
And there's like several types of palettes for you to choose. I always use there's this one's so much better, right? But the default is this color brewer sequential, but you can choose anything. And just a reminder, if you put it green and red, which is very instinctive for lots of people, about 3% of the population won't understand anything because that's the ratio of people who have red-green color blindness. If you put it on blue and red, then about 0.5% of the population will have no idea because that's the percentage of people that have this rare type of color blindness. So usually we always use red to blue because that's the best we can do. That's the most rare type of color blindness. If you didn't have a color scale, you had just like a binary thing, then you could use different saturations. So for example, you have one that is like a pure blue, and the other one will be like a grayish or something like this. And then even if you have the color blindness, you will have a different in tones, and then you will be able to differentiate those. But for color scales, this is the best we can do. All right. So we did the colors now. And another thing that I will try to do now is to go here on tools. And there is this thing called analyze network. It will ask if this is a directed graph. Directed graphs is though that the arrows have a direction instead of edges that are like A connects to B. Is A goes towards B, then you have a direction. In our case, it is not directed, so we can just click OK. And it will create a bunch of other columns. So here on the size, it will say what it created. So it created several different characteristics. And if you click here, I believe it will plot something. It will ask you to install some stuff. But if you install it, it will create a histogram showing the number of edges per node. There will be some interesting things here, but we don't need to do anything right now. So we can minimize this. And now on our node table, you should have extra columns. For example, number of directed edges. Directed is the ones with the arrows. Undirected is what you want. So number of undirected edges. So then you can see here that this line, whatever it is, has 17 edges. So it is for KRT17. Where do you find it? There is an easy way. You go here on style again, if you're not on style, and then you go to size. And size is grayed out because we have here, it says here, check this dependency, lock node width and height. So you can adjust the width or the height, or you can just fix it to a random ratio, and then you adjust size at the same time. This is what you want to do. So here we have a check mark, lock node width and height. So you can put this, and now we can adjust size. And then again, we click here in this middle button, and then it will ask the mapping type. If you put this first and you put continuous, then they will gray out columns that don't make any sense. So for example, the gene names will be grayed out because you cannot have a continuous scale of names. So it might be easier. And then we find here the new columns. Number of undirected edges. And then if you look here, you have some, like the KRT4 is a little bit larger than the rest. And this is how it should be looking for everyone.
Hey, Diogo, we
Yes.
have, I'm trying to replicate things on Web Cytoscape. I'm running into some issues. Well, can we pause here and can you give it a try on your computer to do this on Web Cytoscape?
I
In
could.
fact, just
Isn't it
mapping.
share your screen? Since
If you
you are
could even just import so I can import the network, but I can't get the gene expression results to go in there. I wonder if our students might be having the same issue or past that point.
then source node and target node is a little bit different here. Is it loading? Like for you, it took a while and then okay.
It takes a while, yeah. And you have to zoom out because the node labels are massive.
The default is for you to adjust the width and then the height. But if you go there on the bottom, there's a blue tick box fit custom graphics to node, and beyond that, lock node width and height. So if you check lock node width and height, then width and height will be grayed out and size will be available. All right, okay. And then we will import table from file. And then the important thing is that entries ID should be the key.
Yeah,
It's going to be assigned key. Why is it grayed out? Oh, maybe it is because it needs to be a name, it should be a key. So you say that entries ID is a string, and then it becomes a key. Is this what was the error for you?
yeah. So far,
Now the nodes should have a bunch of values.
yeah, so mine is not, mine looks the same as yours.
I don't know if it's just because it's taking a while. Force table from file straight again.
For our students using the web version of Cytoscape, have you guys been able to load the results table into the browser?
I'm wondering if the problem is that when we did the loading for the first time, and then it considered the interest ID as a number instead of a string, and then the name here is a number instead of a string, so it is not being able to match because it is a number. So when we put number from file, it has here source and target, and it's double the default. So it's probably even replacing 3853 by 3853.0 or something like this. So we need to put here as a string. And then we can try it again. And again, learning from mistakes, whenever you create a new ID, put some characters before the numbers, like go dot dot numbers. So then whenever someone sees the ID, they know it is an ID and where it comes from. All right, so we created a new table. Let's remove the first one to release RAM. And then we try to import data from table from file. Then again is a string and key.
Right. Okay. I think there are some issues with loading data into Website Escape because I'm getting the same thing.
But
But
despite they're not showing
the
here on
style
the
is
bottom,
there.
it has a Gmail
Yeah.
column, and it works. So you have Gnames here. So despite the fact that they are not showing here, you know why? It's probably because one of the table name columns is huge. No, it isn't because of that. No. The web one doesn't work as well, but it works in the sense that we have here the names now, and then if we go to fill color, we have the continuous, and then we can put here the full change. Come on, come on, come on.
Okay, that happened to me too. I think the takeaway here is Website Escape is useful for some exploration, but ideally, we're going to work in a desktop environment. So if you're not able to get Cytoscape working, you can ping us and we can try to work with you to demonstrate the techniques that Diogo is going to lead on the application version. But there are going to be some things that we just can't replicate on the web version. Okay, thank you, Diogo, for that. That was helpful. I wasn't sure if I was missing something obvious.
All right. Is everyone seeing my Cytoscape again?
Yes.
Okay. So, just that so that I can adjust this that's looking so ugly. We go here on size. We double click here to open this window. And so the size was from minimum of 1 to maximum of 8.6 for some reason. It is capping at some position. We can adjust this. So let's adjust it to, for example, I remember that there was one node with 14, so let's put 14 as the maximum. And then, so anything with size 14 will have a 30 pixel size, and anything of size 1 will have a 10 pixel size. And we want to increase this. So if we push it forward, it will go to ridiculous amounts. So we can put something like this, so that it becomes very clear which are the main ones. Let's close it. And did it go back? Okay, something happened. Oh, I closed it without pressing OK. Was a rookie mistake. Okay, so now
So
it's there.
what does the min and max are representing here?
Is it scale? So let's open. So here, for example, the min and max is 4.27 because that's the maximum value that they found. But we can set min and max to different values. So for example, if we put here -1 to 1, then all the colors will be so much more drastic because they are reaching the limit, right? Let's return to where it was before, -4.
So it rescales
Yes. So in
everything?
this, yes. So if I click reset here, it will put the maximum and the minimum. And if you have an unbalanced one, it will be kind of strange. So I can say, oh no, I don't want it to be imbalanced. I want it to be symmetrical. So I put here +4 and -4. And then nothing will be completely red because the scales now don't go to the end of the red. In this case here, the default of maximum was less. I don't remember which was. I believe that's 8. So now all of these large nodes are large and they aren't different from each other. So this node here in the bottom and this node here in the top, both of them have a size larger than 8. So it will be kept at 8. So this will be quite useful if you have like one that has 1,000 points and it would dominate completely the figure. So it is important to have minimum and maximum. But let's put also the maximum is 39 apparently, which is too much.
So it doesn't rescale, it's thresholds, right?
Yes, this is the threshold. And then if you want to change the scale, you can do it like this.
Okay.
And you can also like do some crazy stuff. So I know that you can add another thing in the middle so that you have like a janky thing. How is it here? Sorry, I don't even remember. Oh, here, add. You can add new points and do some strange things. Let's not do that. That is very counterintuitive, but it's something that you could do if you need to. Please return to where it was before. Yes. Okay. So now our mapping works. One thing that sometimes you can do, you can press Shift to select some things, and you could, for example, press delete and remove those that are lost. You shouldn't do this in a publication because would be considered like faking your data because you're removing some points manually. But whenever you're like editing images and things like this, sometimes you just want to take them out of the way. You could move them around like this. You could remove them, delete them, whatever. So let's just move them around. Select the main ones, and then you go to layout here. And there are several automatic things that you can do, like circular layout. And then it becomes like this. It puts everyone in a circle. I said those guys that have only one connection. You can put, there are many types of layouts. I really like organic. This is usually what people expect. So it will try to more or less keep them aligned in a regular distance to each other. And there is another one that is quite interesting. There is this edge weighted force. So it will basically predict every edge as a spring, and then you can put a value to it. So for example, in our case, we don't have any interesting characteristics. Oh yes, when we created the analyze network, it added some more columns to the nodes and some columns to the edges. In this case, it added to the edges the edge betweenness, which is this measurement of how often the average path between 2 points pass through this edge. So we could use this force directed one. So the force will push them together and the spring will push them apart or something like this. And then we put on the selected nodes, and then we can put edge betweenness here. And then it will create this network in which let's put it aside a little bit, in which the weak links, so for example, this link here is on the periphery, it will have a different size than those links here in the middle. Like this guy here is quite central, so their links are smaller. So this is different ways that you can deal with clustering. I usually like using organic layout. And then you can also do this thing, edge router. It takes a few seconds, not many. Maybe it's taking too long because I clicked on the orthogonal edge router. I wanted the organic one. Organic edge router. All right, not that many seconds. So it created these knots in the strings. So now you can see that, for example, this guy here is going around the edge. So they are not passing beneath each other. So you can see that they curve here. So this link here was supposed to go behind the nanog, and now it's like going around it. So this is a neat thing to make things easier to follow. So before we had a monster that was impossible to see anything, and now it's starting to have something more tame. Now we have something that, okay, it's still messy, but we can see here that there are these central nodes. If they are central, they are here because they are more important. And then you have the sizes as well. So some guys are still kind of lost. But you can see that by the sizes, which are the important ones, and then we have the colors also. So then, oh, look at this guy. This guy has a very large fold change. It's not super important though. This guy here it's not a deep of a blue, so it's not that much fold change. But this seems to be more important because it has more edges. Therefore, it is a protein that interacts with more proteins, and it is more central. So then it should mean something as well. So these are different ways you can add more dimensions to our plot to make things easier. Let's see. All right, okay. So while I am doing this stats review, guys, I'm following the slides that I published. By not looking at the slides right now, you're not losing anything. It will be exactly the same thing. The difference is that the slides are here for you guys to follow if you are like at home and you want to redo this later, then you have the slides to follow. But I'm doing exactly everything that is on the slides right now I'm saying, so you don't need to follow them right now. Oh yes, and of course, as I said, you can zoom in and out with this. In we can use Shift to select some folks and move them around. And sometimes this is not too much of our case. We don't have many clusters here. But one thing that we could do, for example, if we have like, oh, sorry. If we have a cluster of genes that is more or less separated, we can push them apart, and then we can reorganize them. So, for example, here, if I click here, I believe it will do them all. Huh, no, yes. So they push it together, but you see that they reorganize the organic just on this subgroup. So if we have, for example, the circular layout, no, it changed everything, sorry. But some of those you can allow to just put the selected nodes only. So here, for example, let's put just the selected node ones and organize them in a circle based on between the centrality. And then you have this circle here. And in this corner here, there's this important tool that looks like a constructor L. And we can do this. And then we can select a bunch and then push them closer together or further apart. So I'm doing Control Z to undo my mistakes here. So if we have here a bunch of things that we want to, like, oh, I want to expand this middle, for example. So I can select everyone, unselect the middle. Oh, sorry. Oh, sorry. You select these guys here. Every time I click on something in the middle. And then I go to selection, and I can do an invert selection. Nodes. These are interesting things. For example, first neighbors, I will select whatever I'm selecting plus their neighbors, for example. But I want to invert a node selection. So invert everything but the center. So then I can go to the tools here and increase them. So I can more clearly see this differentiation. And sometimes also, I might want to turn them around. So let's do it like this. Edges or nodes, sorry. Invert selection. You can, for example, rotate everything. In this case, it doesn't make too much sense to rotate them, but I will show further away an example where it will help a lot. Okay, so this is what we can do with any list of genes. So if you don't have the data of their connections, you can take connections from other folks like BioGrid. So this will be like these proteins were seen somewhere doing this kind of connection. If you have data from positive protein interactions or things like this, you can create your own connections, and then you can say things like, oh, in lungs, because I'm analyzing lungs, these proteins are known to connect to each other. So then you have a more strict number of connections, and then you have a different network. Let's now try to do the nodes with pathways. So when we go back here to the code I sent you guys, There is a small chunk of code here in the bottom. So I'm taking this file here, gprofiler results full change 0 ordered, that we created last class. This is so when I do it like this, just so that you guys see what we were talking about. These are the results from gprofiler. So we have here the name of the terms. Small molecule metabolic process is the first one. We have the p-values for enrichment. Somewhere here is the adjusted p-value. Anyways, we have term size, we have intersection size. Apparently, we don't have the FDR, but we don't need it. The important thing is that we have here the intersection. So this is the list of genes that are significant. So with that, we can try to create those nodes that are connected by the fact that they have an intersection. We need, therefore, the list of genes that make it. So you can go to the website of Enrichment Map, and you can see on their manual how the input should look like. They have an explanation there. But in summary, it is just a GMT file. So we have the term ID, the term name, and then the intersection. So we can create a new one just with our pathways. So if we do it like this, we will select from our pathway just these things. Oh no, this is very long. But here we have these lists of genes that go forever. And unlike in here where they are comma separated, they should be tab separated. So there are many ways of doing this. In this case, I'm going to use this function called gsub that will be a global substitution, and we will replace all of the commas for tabs on the column intersection. So now you have tabs on all of those. And then we can save this file as a text file. And then if you go here to outputs, I called it mini GMT. So you can see here term name, term ID, intersection. So ID, sorry, name, and then the intersection, right? So remember that we downloaded GMT files from the sources, like gene ontology and KEGG. So KEGG not, you cannot download from KEGG, but Reactome. You can make an enrichment map with all of those. It will be absolutely giant, but you can do this. In this case here, we are selecting our networks, and we are selecting just the differentially expressed genes. And then we go back to Cytoscape. Instead of creating a new table like we did before, we're going to use this tool. So here on apps, you should have enrichment map. If you don't have enrichment map, you will need to go to the install from App Store, and then you have to find enrichment map. Searching here, enrichment, and then it will be here, and then install it. But you should have already installed it from the pre-work. How much time do we have for the end?
We have an hour.
An hour. Okay, so I'm going to census here. How many of you have enrichment map working? So if you do, you should have it like this. You go to apps, enrichment map, and then you should have this screen. How many of you don't have this screen? If everyone has it, I'm going to continue. Okay. So all of these apps will eventually do something, right? And usually this starts opening the screen so that you can load the files. So just like before we did the import from file and then we put a file here, we'll do the same thing. So in this analysis type, they have a GSCA, and then it will receive those RNK files and other files that we prepare for GNCA. You can put them here. I'm using this generic one. And then you can see that the asterisk asks for a name, which is just a name, it's set, and then it only asks for a GMT file. If you put ranks, expression, classes, whatever, it will try to color the nodes accordingly. It will try to do something for you, but you really need is the GMT. And then, so again, we click on to add a new one, and then it should appear this analysis type. You can put generic. And then here on GMT, you can put the file that we created recently called mini GMT, right? There are some options here on the bottom. The most important one is this slider here in the corner that goes from sparse to dense. We could change it before, but we can change it later too. So let's change it later. Do you guys have this on your screen? Is it working for you?
Yes, it works.
Yeah.
Cool. So we have this. And again, this is kind of crude. It is a little bit hard to make sense of everything. We have too many edges here. Look at this mess. So we go here to enrichment tab, and we have these cutoffs here. Remember that I was talking about the Jacquard index in the past. There is a ratio between the union and the intersection. So in this case here, we can use this slider here. And as we push the slider on, to remove some of the edges. So it will remove edges that aren't as important, basically. So if we do this, it will be too sparse. We have too many loose guys. But there is, you need to release the mouse to recalculate everything. So we can go here and choose with, we have a few connected clusters like this. Oh, too many. So I believe that 0.64 was the best one. I'm checking just 0.64 because then it keeps this connection here. Maybe we can actually take all of these nodes. So let's put a little bit more so that we can have these guys connected as well, like this. So 0.55 is the threshold that I chose. Okay. And now we can select everyone. Actually, we don't need to select everyone. You can just go here to the layouts and go to the organic layout again. And then it will reorganize it based on the edges that we removed. So now we have these clusters here. They are very intense. We can push them a little bit to the side. So oftentimes you see in publications some nodes that are separated like this, and then we can
for the interpretation, we'll based on just the color or the node and the edge for the interpretations, or
in this case here, I'm using just the edge structure to create these separations. We can increase this just to make it more readable. And sometimes you have 4 nodes aligned like here, so it's cool to rotate them so that they are not one on top of the other. So when I do it like this, the names don't overlap as much, so it becomes clearer. And we can add colors as well. So just like before now, we can import.
So for the publication, can we only publish clusters, not the whole thing?
So one thing that I did already in the past is to show something like, oh, I have here a list. This list created 6 clusters, and I'm only showing the largest 2, for example. So then you only show this guy, and then you remove the rest. So you can do this.
Okay.
Usually, if you do this, I would recommend that you put on a supplementary figure the full thing that is giant, and no one needs to, it doesn't need to be pretty. So you need to have the data somewhere so that no one says, oh, you're cherry picking your data, or you are being misleading because you're removing things that don't agree with your thesis. So some people will be critical of you removing things, but for clarity, it makes sense. You just need to then be careful to let it also be available for those that are more picky and want to see it all.
Sounds good.
So, is there a way to save a particular cluster, or can we just do that eye icon, just make it invisible and then save it as a image file?
Good question. So when you save, you can save session. Like lots of people have used Photoshop, and you can save an image. But if you keep the Photoshop file as it is, you have all of these layers, you have other information that you lose when you save the image, right? So if you can just save session, and you create these .cys files. And then you could save one, and then you can remove the nodes, and
Okay.
then you can save another one. So let's just take this. I removed everything already? Okay. I don't know when I did that, but let's remove this guy as well. And then we can save again. And then we have here favorite cluster. And then we can focus on working on it, and then we can go back and we didn't lose everything else.
Okay.
And of course, sorry, you can export as a network to image, and then it will have some things for you to choose here. PNG is a choice you can make, but PDF is a little bit better because it will keep it vectorized. So sometimes you have an image that when you zoom in, you see just the pixels and it gets too blurry. And in this case, for example, I can zoom in forever. That's the difference between a vectorized image and a pixelized image, or rasterized is another name they give. So if you save as PNG, you zoom in infinitely. Eventually, these lines will be jagged because they are pixels. But because we are on this raw format, it knows what a G looks like, and it will generate infinitely the zoom, so you can zoom in forever. So it's good to, up until the last minute, keep things in this format, and PNG respects this format. So you can save things as, sorry, PDF respects it, PNG doesn't. So if you're going to export, I recommend doing it as PDF. But yes?
No, I just said thank you.
All right. Excellent question.
I had a question. If we run clustering, does it generate clusters similar to these clusters, or it will be different?
There is a random randomness to it. So the organic, for example, if you run it several times, you see that it changes a little bit.
Because I remember we could have different clusters in different subtabs. So you didn't have to save the session. You basically could have different clusters in different tabs.
So what you did was probably better than what I was doing. As I was doing these examples with you guys, I started with the genes, and then I decided to add a new thing. And I didn't close Cytoscape and open it again. So I have here on network, I have here the original. Sorry. Come on. I still have here the original thing that I did 20 minutes ago. So this is in this network tabs here. So yes, if you are clever, you can put each of the clusters in these tabs, and then you don't need to save a file and reload it. It is a better thing that I was doing indeed. Yes. So let's go back to what we were doing before. And of course, there are different layouts you can do. This will, when you said different ways to cluster, I thought you were referring to that. Another thing that I skipped is that just like you can cut off the edges, you can cut off the nodes. So by p-value. So you have somewhere here on the node table the p-value here, enrichment p-value. So you can go here to enrichment map and say, oh, I have too many nodes. You know what? I just want to have those that have a significance of at least 0.001. And then I remove everyone. Okay, 1%. So then this cluster here is more or less intact, but the rest was removed. So when we did yes, so let's start it again real quick. Instead of loading the file, we can do it like this. Mini GMT build, and then we will create a new one with all of the nodes. And then we will choose them instead of just by edge connectiveness. We'll first filter them. Oh, let's add color as well so that we can have an even more informed choice, right? So you go here to import table from file, and then we have our original file here. Ensemble ng, gene profiler results, flow change 0 ordered. And then we have the key on the, I don't recall, yes, we have ID. So this will be our key. And then when we go to the nodes, we have all of this information. And then we can go to style and color them. Let's remember what I colored this with last time. Oh yes, we ran Pathway Nester last time. So I also added this one. So let's create a new table here. Import table from file. And then we put this one that has filtered gprofiler results for change ordered. If you don't have this file, you don't need to just put the color. Actually, let's simplify our work here. Let's not add this one too. And we can color by something else. So here on filter, sorry, not filter, style, color, fill color. And we have a bunch of, yes, that has p-value, right? So there's a bunch of columns here that came from different places, I suppose. in our case, what's the name of the column? Precision recall, termite. There's something wrong here. You see that the term name is the ID. This has happened when R has column names, and then it will shift all of the labels forward. So the labels here are wrong. So let's just open this with Notepad. And then we can see here, I'm gonna zoom ridiculously. There's query one, significant, there is one. And then we have this one here, this is the line number. So sometimes R does this thing where the line number doesn't have a column name. So then we can put just here like a number. This isn't like necessary, right? But just to keep things organized. And so I saved this. I lost my mouse. And then we go here to Cytoscape and edit again. And then the column names are correct. I should have noticed it before. Oh. Ooh, there is something wrong still. Source should be GOBP, and term name is here. So there is a missing column here somehow. Okay, we may not fix this right now, but at least we know that this thing called term size is actually the full change. As I noticed before, we were missing the FDR, so maybe somehow we lost it from the header. I don't know, but let's consider that this term size is actually the coloring that we have. So we put here term size. I changed the name of the table, so just let me term size exactly. So then fill color. Because we are somewhat in a rush, I will not go back here and figure out why these titles are mismatching. But oh yes, and everything became white because it's a p-value, so everything is from the closer to zero, the more significant it is, right? So we need to change here the current mapping. And then we need to put it in log scale. How's the maximum 2? Oh, this should be the maximum, okay. And let's invert the colors here. So we put in red the things that are most significant. And then in white, the things that are less significant. All right. So now we added colors to the nodes. Again, if we had more time, we could do it with more care. We could do it with more care. We could do it in log scale properly. In this case here, I just putting things closer to zero, right? And then we have our nodes here, and we can go to enrichment map and do a full change cutoff. So we have too many nodes. We don't have time for this. Let's only show guys that have at least 0.05, so 0.5%. The nodes are as we were saying, the columns are shifted somehow. So we have to do it more manually. But we can, as I showed before, select here on the node table. We can organize them by term type, which is actually FDR, as we know now. And we can, so the largest one is 4%, 4.9. So we can come here and reduce them, select with shift, and then we select nodes from selected rows. You see that they will be selected here, and then we can delete them. Press and delete. Select nodes, delete. Remove selected nodes. So when I was pressing delete, it was trying to delete from the table, and I had to click here to go delete. But when I click it, it didn't select it. So I did it like this. Sorry for the mess here. Something happened with the headers, but were you able to do what I did right now? You select the from term size, you select up from a certain value to a certain value. Let's say 0.5%. So if we want to delete more stuff, for example, we come here, we select those that we want. Go here to select the nodes from selected rows. They will be yellow. And then we can, for example, move them around, or we can delete them, press and delete. Or if they unselect, you can also try with the edit, remove selected nodes. Did it work for you guys? So I will redo everything that I did. Organic layout. I'm gonna remove these terms that are not too significant. Let's have a 1%. Select nodes. I'm going to delete them. And then we can also change the amount of similarity here. I was putting 55 this time, right? 44, 45. And then we can, for example, let's move this guy aside. And let's work just with this one here that is more interesting. I'm saying that this is more interesting because we have this structure that we have a bunch of guys clustered together, and we can say, oh, these can be grouped together as a whole. And on this side here, we have some other ones that are, we have some other ones that are connected by like a bridge. And then these ones we might take a special attention to. So then we will rotate them around, zoom in a little bit. And then we have these guys here that are a mess. And then we give a zoom in just for them. If you zoom in and select zoom in, select zoom in, select this thing will go to the corner. And then you just click on these arrows to renormalize the then we rotate it here, push it around. All right. Now we can start to see sensing things. It will always require a little bit of manual editing. But here now we have some clusters that make sense. And then you can see here that the edge thickness comes from these enrichment map variables. So similarity coefficient, I believe, is what defines the witness. We can also change this if we want, of course. Here we have all of the node qualities. On the bottom here, we have the edge qualities. So the thickness here is width. Here, the last one, not width, thickness is width. So I can adjust this as well. So I can say that I want the larger one to be more noticeable, for example. Then we can change here the maximum and minimum. And then it will increase the contrast, for example. We can edit here. Um, screen here, based on everyone else is trying to having the same problem as me.
I got the same as you, Diego, and then it brings up some sort of a jar file.
So the jar file should happen when you open App Store install from file.
Yeah. Oh.
If you go to the show App Store, this time it appears here on the side. I don't know why, but usually it appeared for me here as a separate window.
Yeah, mine is also on the side tab.
Can you look for auto annotates there on the search?
Yeah.
Look for what?
Auto. So it's here like
Auto annotate. Okay.
Auto annotate with two ends, everything combined. And I click on search. It kind of freezes. I believe that maybe it is trying to open Firefox to have this window here.
Yes, when I search auto annotate, it will open a browser to auto annotate to install that.
Yes, all right. So this auto annotate has like less than 1 megabyte or something like this. So it is super quick to install. Then it downloaded this jar file. It says here that the app is part of an enrichment map collection. So whenever we sent the link to install an enrichment map on the pre-work, maybe we should have sent the collection. This is why it wasn't there, sorry. But now we have this jar file that we can install. Install it from file. Then we go to the downloads. And then we select the auto annotate. All right. Let's just save just in case. And then we can go here to auto annotate on the apps. My laptop is not handling this very well. So then it will ask here, what is your label column? Since our columns were shuffled around, we need to make sure that the annotations are on the correct size, correct place, sorry. So on the nodes table, there's a message going on. It's called GSDisRF. Should I click on the lock node checkbox? Let's find out. The annotation set. I don't have this checkbox.
No, that was for before when I didn't have this size activated.
Oh, all right. Did I receive a recent message then? I have the select notifications going on here.
No, it is just like we asking like if we need to arrange any breakout rooms.
All right. Sorry. I think this should be very quick to the end because we just click here. As I said, you can adjust the amount of clusters. Let's have more clusters a little bit. Layout networks too. There's advanced with like maximum words per label and things like this. We can adjust this later, right? But the idea is that, for example, if some words appear several times, then we can limit, like, oh, I only want words that appear several times to be here. So then we can increase, for example, this minimum word occurrence. And let's create annotations. Okay. Where my computer is screening here. So it created here some automatic annotations. Because I thought that we only had this pathway in the middle, it created this huge cluster here. But because I didn't remember they had other clusters here. So, but you can see here the way that it works, right? So it says that this cluster here has acid catabolic process, which is just a mixture of words that came from this mass inside here. So we need to check that it makes sense. Extracellular matrix collagen, like in this is a little bit more sense because the collagen matrix is usually extracellular, right? And we can see here where it took the names from. So here, these things here in the corner aren't extracellular at all. It are collagen or matrix. So ideally, we should remove them from this group. So whenever we were running auto-annotate, where is it? Auto-annotation. We can change here the number of clusters to have more clusters and separate them more. But here, this corner here makes more sense because we have here a central node called extracellular matrix organization. We can see here other matrix extracellular things going on here. So this node here makes more sense. So when you're doing auto-annotate, we need to be very careful with what you let the algorithm do, because this is not something that I would consider acceptable, because it's considering that these guys are on the same nodes when they shouldn't. So we actually should need to increase the number of annotations. So we go to the options here. Sorry. Yes, we want to increase the number of clusters, and I'm not being able to edit this here. Okay. Can I start? We start off. So let's start it again. Sorry. Auto-annotate, the annotation set. And then here on amount of clusters, we want to have more clusters. And we can also remove the data sets that we don't want. So all right, that's better. And now we can see here that these were relabeled development, which is something that makes more sense with this cluster. And then we have this larger cluster. It was subdivided into smaller ones. So we can push them aside here and check what they are doing. And clearly here we have too many clusters. So this one here is too much. Oh, we removed the wrong one. All right, sorry. When I do undo, it doesn't undo the changes that I made on the cluster, just undo the changes that I made on moving them around. Connie, you that also used annotator before, do you know what I'm doing wrong here? When I'm trying to redo the annotation, I can like delete and create a new one. But if I just want to change the number of clusters, there are more clever way of doing this than redoing everything from scratch.
I don't have a more clever way. I usually end up redrawing.
All right, okay. So let's do it like this then. We delete. And we start again. A little bit more, a little bit less now. Create annotations. I'm pretty sure there is a way to redo this without having to delete everything and do it again. But I'm not finding it. Sorry, guys. It's been a while since I don't use Autonate. Actually, when I use Autonate, I never had this problem before, so I don't know why this is happening. But this is the point. We can use Autonate to create these sets, and there are some adjustments to be made, and that is the important thing. All right. Let's have the break now.